Skylar, Eric, and I did our macro presentation project on the lac Operons and fatal familial insomnia. So what is fatal familial insomnia, or FFI? FFI is a rare genetic prion disease caused by a mutation in the PRNP gene that mainly affects the thalamus, causing the production of misshapen or misfolded proteins. FFI is a progressive neurodegenerative disease characterized by the inability to sleep or insomnia that may start mild but will progressively worsen, leading to severe physical and mental deterioration and death. FFI inheritance is autosomal dominant, requiring the inheritance of only one allele. Prions are, for those of you who don't know, are misfolded proteins with the ability to transmit their misfolded shape onto normal variants of the same protein when they come into contact with one another. The PRNP gene is the human gene that provides instructions for the production of the PRP protein, the prion protein. This is a gene that we all have. The PRP protein is active in the brain and other tissues throughout the body. The exact function of the PRP protein is still unknown, but researchers have proposed its function may be linked to cell signaling and in controlling the circadian rhythm. The PRNP gene is a gene we all have, and when a mutation occurs in this specific gene, it can lead to the genetic disorder FFI. The cause of FFI has been identified as an autosomal dominant mutation at the codon 178 of the PRNP gene. The PRNP gene is found on chromosome 20 at position P13. A mutation on this gene causes the PRP protein, the prion protein that is made from this gene, to be folded incorrectly, resulting in changes in the thalamus, including the progressive loss of neurons. The thalamus functions in relaying messages between different parts of the brain, managing sleep-wake cycles, the flow of visual, auditory, and motor information, as well as aspects of learning, memory, speech, emotional experiences, and expression. This loss of neurons in the thalamus causes many of the symptoms of FFI because the thalamus then can no longer function properly. Although the main target of FFI is the thalamus, other parts of the brain are affected as well. As you can see in the photo to the right-hand side, it is a PET scan of a normal brain on the top and a brain with FFI on the bottom. And you can see that everywhere, really, there's this appearance of less kind of activity in the brain, but specifically in the thalamus. The first symptoms of FFI usually begin between the ages of 32 and 62. However, with a mean of like 51 years, kind of differs person to person at the onset of the disease. Many symptoms of FFI are similar to symptoms of other progressive neurodegenerative disorders such as Alzheimer's and Parkinson's. It is important to note, however, that insomnia is not always the first symptom of FFI. Sometimes the first symptom is progressive dementia. When insomnia begins, it usually comes on suddenly and steadily worsens over a period of a few months. As the disease progresses, most people with FFI develop abnormal uncoordinated movements, hallucinations, severe confusion, delirium, and muscle twitches and jerks. And that is directly the result of not being able to sleep. When you can't sleep, you start having hallucinations and this severe confusion and all these crazy things start to happen. Other symptoms may include panic attacks, phobias, weight loss, lack of appetite, and having a body temperature which is too high or too low. Late stage FFI, while the dementia may start as forgetfulness and confusion, it eventually leads to the inability to walk and talk. While insomnia starts as a difficulty going to and or staying asleep, the complete inability to sleep is quite common towards the end of the disease in late stage FFI. This complete inability to sleep is fatal. FFI typically causes death within 12 to 18 months from the onset of symptoms. Sleep is as essential to life as food, water, and oxygen. Here we have, to the right-hand side, some EEG brainwave patterns during different stages of sleep. So on the top, we have, for a relaxed wakefulness, these appearance of these alpha waves when we're awake. And in stage N1 or light sleep, we have these theta waves. In stage N2, we have the appearance of these sleep spindles and this K complex. And deep sleep, stages 3 and 4, you have delta waves. And it's these delta waves that late stage FFI, where they kind of just stay in that dreaming sleep REM state, they never get those delta waves because they never reach, they're incapable of reaching stage 3 and 4 or a deep sleep. So those delta waves don't happen because they never get to that stage of sleep. They kind of just exist in a weird space between REM and awake. So not fully awake, but not fully asleep.
Okay, so for our background, for our experiment, we looked into different papers that talked about FFI. This one talks about FFI in mice. So the title of the paper is Transgenic Fatal Familial Insomnia Mice, Indicated Prion Infectivity, Independent Mechanisms of Pathogenesis and Phenotypic Expression of the Disease. So in simple terms, pretty much they were studying the sleep disruption in the mice that carry this gene that Keely had just mentioned. That would be the mouse equivalent of FFI. And how they studied was they used the transgenic mice and put the PRP gene mutation in them. And how they studied these mice was with a sleep-wake behavior assay and an EEG, in addition to a bunch of other assays. But for the purpose of our project, these are the two assays that we focused on. For the EEG, they simply just anesthetized the mice and did a regular EEG on them. And then for the sleep and wake assay, what they did was they sleep deprived the mice, and they did this by gently handling them for about 6 hours, and then they were put back in their cage, given food, water, regular standard conditions, and then allowed to sleep and behave as they regularly would for 18 hours. Okay, so the figure I chose from the paper shows REM sleep with 2 EEGs. And as Keely had previously stated, REM sleep is when the body is paralyzed and then kind of dreams begin. And on it, it shows A and B, which is mouse A. That is the mouse that is going to show FFI, and B is the normal mouse that it's compared to. And as we had previously learned in class, EEG showed different brain waves, and Keely had showed during the different sleep patterns. So this showed their different brain activity. The arrows shown in the figure point to the REM sleep specifically. And the black lines on the EEG on the bottom two, show the delta waves, which are high amplitude waves and are common in deep sleep. And then the red waves are theta waves, which are considered kind of slow activity. So, and then where it says NREM, those are the sleep stages 1 and 3. They kind of group them all together. So that's all the stages before regular REM sleep. And pretty much why this figure is significant is that it shows the disturbed sleep stages in the mutant mouse, kind of how Keely said, they kind of stay in this in-between of REM sleep and wakefulness. They don't really go through the stages as the normal mouse does. Okay, and then kind of just to sum up overall what the whole paper found, the mutant mice showed the same sleep abnormalities that humans with regular FFI showed. which is what they were kind of hypothesizing to find. And then they also found that the same abnormalities in transport proteins were found. And they also found that protein aggregation was causing these selective neuronal dysfunction.
Okay, so our study questions, what we're looking at and questioning is, we're going to look at the effects of turning on and off the mutant PLP gene in mice before and during the onset of FII, and the effect that has on the mouse's sleep cycle. Does it make it better? Does it make it worse? So it's supposed, as previously stated, the mutant PLP gene produces proteins that have neurotoxic properties and result damage to the thalamus, causing it to suck into sleep. The question is, if there's a disruption in the mutant PLP gene itself, which results in it no longer being able to produce the mutant PLP protein, will that stop or reduce the damage to the thalamus? And then will this result in a more normal EEG for the mice? So overview, what are we going to be doing? The mice will contain the human PLP gene with the lac system inserted upstream of it. It will contain both the unmutated gene and the mutant PLPN gene that causes FFI. The suppression of the PLP gene will then be controlled by the amount of lactose present in the mouse's diet. Since the brain usually runs off glucose, this means that it will, unless we increase the amount of lactose in the mice's diet, then it will always be off. So this is a good thing. It's easier to control. There will be 4 groups of mice present. The negative control, so that's the unmutated mice given a regular diet. This is the base control, nothing changes. Positive control, mice containing the mutant human PLP gene that causes FFI. And then the control, mice who have the non-mutant PLP gene, but they still have the on and off switch upstream of the gene. This is just to ensure that the on-off switch does not cause any issues on itself, any alterations. Mice who have, and then finally the experimental group, which are mice who have the mutant PLP gene and the gene upstream of the mutant gene. So quick little recap on the lac-operon gene. Lac-operon gene is a promoter site and a repressor site. This inhibits the transcription of genes downstream of that system when in the presence of glucose, but it promotes the transcription when in the presence of lactose. So that is how we'll be turning the gene on and off. So the organism we chose is, of course, the common house mice. Now, why did we choose this? Mice who have, one, the mice have already been shown to been created that have the mutant human PO gene. So that's great, as previously stated in the first paper. It's a common human analog. We use it a lot, especially for studies to compare structures in each other. It has an advanced neural network similar to that of humans, which is required. We can't really do this experiment on bacteria or C. elegans since they do not have a similar nervous system, and they have relatively short lives, so we can actually see FFI progress in a relatively short time frame. Methods. So of course, we'll be using CRISPR to genetically engineer the mouse to have a single allele of either a mutant human PLP gene with the lac system upstream of it, or the normal human PLP gene with the lac system upstream of it. Then to determine that they are producing the either mutant or non-mutated form, we'll use a Western blot analysis to identify the mutant PLP gene mice and producing mice and the mutant PLP protein in both sets of mice. Then we'll use an EEG to study the sleep patterns of the mice. So 3 groups. Mice with the human mutant PLP gene will be placed into 3 groups after they've been identified using the Western blot analysis. Group 1 will have the PRNP gene turned off once they reach adulthood, so around 3 months. Group 2 will have the PRNP gene turned off once they begin to exhibit signs of FFI. And then the final group, Group 3, will have the PRP turned off since birth. So this is just to determine if there is also any negative effects to not having this gene turned on. And then a sleep study will be done with all groups, as previously with the controls. The anticipated results are mice with the mutant human PLP gene will not develop FFI if the gene is turned off since the birth of the mouse. Mice who have the mutant PLP gene turned off during adulthood will not develop FFI, or if they do develop, it will be later on in the life cycle, as the as the mutated prion proteins will not have as large of a density as they were not constantly being produced. Mice who had mutant PLP gene turned off once they began to show symptoms of FFI will continue to have FFI, but will proceed at a decreased rate because the protein is not constantly being produced.
So in conclusion, kind of what the significance of this experiment would be and why the research needs to happen. This could possibly lead to the development of treatments for FFI in people because currently there are no treatments just because once the symptoms start, it's a pretty quick progression just to them dying. And so if this did lead to treatments being developed, that would greatly improve the lives of FFI patients. So they could potentially keep living and not just die once their symptoms start. And obviously that would increase their lifespan. And another significant aspect of this would be that it would lead to much more research and discussion on the topic. FFI is not very well known and there's not too much research on it. If there was more research on it, that could also lead to the development of treatments.